Hello and welcome to the video on Human Genome Project. Human Genome Project was a project that aimed to sequence the entire human genome and provide its data to the world. So basically there was a global collaboration that happened, and this was the largest biological research project that was ever undertaken, and it involved like thousands of staffs and institutes across the globe. After it took 13 years for them to find a rough draft of a human genome that was published in 2003. It took them 13 years to make a rough draft of a single human genome. Right, so the genome is a chain that's very long. It might stretch up to almost 2.5 kilometers if you took it out and put it in a line. It currently provides information of 3 trillion base pairs and sequence of around 30,000 genes. The data that is being made out of all these research is given to free and open access to everyone in the scientific community, and also the normal public domain can also be use it for freely and using online public databases and all. Now, what are the applications of this? Why was such a big project undertaken? So we all know that gene is what makes us our all characteristics, like how tall we are, how short we are, etc. So basically, if we have some problem, and instead of taking a medicine, if we could alter a gene itself, how cool would that be, right? So basically, to treat some diseases, scientists thought that if we knew the sequence of a healthy genome from a healthy person, then we could alter the gene of a sick person. Like for example, if let's say A and G are supposed to be together, but instead of A and G, that is like we told in the previous video, there's A and C, for example. Then in the sick person, then we could alter the gene to make it A and T to correct it. So the applications are first of all molecular medicine, right? So through genetic research, medicine will look more into the fundamental cause of the disease rather than concentrating on the treating of the symptoms, like I told you. And the genetic screening will be enable rapid and specific diagnostic test making. It is which makes it possible to treat countless maladies, and the DNA based test clarifies diagnosis quickly and enables genetics to detect carriers within the families. So if there's a family hereditary problem also, that can be fixed by altering the gene. Since the gene is coming from your parents, and if they have any hereditary disease such as diabetes or sugar like that, so that can also be identified, and your gene can be altered to a healthy person's gene, and hence your offsprings will not have the hereditary problem, right? So the genome information can indicate a future likelihood for some diseases. Now then comes waste control and environmental cleanup. So basically, microbes that live under the extreme temperature and pressure conditions have been sequenced. So we know there are few bacteria or molecules that we know, like microorganisms that can live up to very cold places and also in very hot places. Like we had recently discovered that there are bacteria under the volcanoes also. That's so hot, right? That's in under the lava. And also there are bacteria that are present in very cool temperatures like Antarctica, etc. So, microbes that live under extreme temperature and pressure conditions have been sequenced. And by learning the unique protein structure of these microbes, researchers may be able to use the organisms and their enzymes for such practical purposes as waste control and environmental cleanup, etc., to degrade the materials. Then comes energy source. So, having the genome sequence of the methane producing microorganism, will be allowed the researchers to explore the process of methanogenesis. That is, in more detail, we could lead to a cheaper production of fuel grade methane, right? Now we have seen that there's a lot of shortage of fuel since the fossil fuels and all are getting over, right? So this might also give us a chance to make fuel grade methane. Then comes risk assessment. So the ability to access risks posed to individual by environmental exposures to toxic agent can be identified. So scientists know that the genetic differences cause some people to be more susceptible than others to such agents. Now, to understand the effect of low-level exposure to radiation and other energy-related agents, especially in terms of cancer risk. So one major application is that it helps with cancer treatment, right? So you can like try to alter the gene of a cancer patient, and you can study many things using this. Like let's say in Africa, people are more immune to like diseases than compared to in the northern hemispheres, like the America and all. They are very less immune, right? So why are these Africans more immune than the Americans, and how can we make the Americans also more immune, right? For example. So this is all about the Human Genome Project. Thank you.